All right, 15.2, recombinant DNA. Kind of is what it sounds, recombining DNA. So the goal is to evaluate the impact of biotechnology on the individual, society, and the environment, including medical and ethical issues. The far side says early man. Do you get it? Do you get it? Do you get it? Maybe not. All right, take a close look at that one. Objectives are to explain how scientists manipulate DNA, describe the importance of recombinant DNA, and define transgenic and describe the usefulness of some transgenic organisms to humans. Let's think about it first. Suppose you have an electronic game you want to play. You want to change. And the game depends on a coded program on a computer microchip. You need a way to get the existing program out of the microchip, read the program, make the changes you want, and put the modified code back into the microchip. What does this scenario have to do with genetic engineering? Well, pretty much everything. Our objective first is to explain how scientists manipulate DNA. Until recently, plant animal breeders could only work with variations that already exist in nature, and the changes produced by mutations were unpredictable. Today, genetic engineers can transfer certain genes from one organism to another, designing new living things to meet specific needs. It is relatively easy to extract DNA from cells and tissues. The extraction of DNA can be cut into fragments of manageable size using restriction enzymes. These restriction fragments can then be separated according to size using gel electrophoresis or another similar technique too. So in 1987, Douglas Prasher, a biologist at Woods Hole Oceanographic Institute in Massachusetts, wanted to find a specific gene in the jellyfish that coded for a molecule called green fluorescent protein, or GFP. This natural protein found in the jellyfish absorbs light, energy from light, and makes part of the jellyfish glow. Prasher thought that the GFP from the jellyfish could be linked to a protein when it was being made in a cell, a bit like attaching a light bulb to that molecule. To find the GFP gene, Prasher combined part of an amino acid sequence of the GFP protein to a genetic code table and was able to predict a probable messenger RNA base sequence that would code for the sequence amino acids. Next, then, Prasher used a complementary base sequence to attract an mRNA, a messenger RNA, that matched this prediction and would bind to that sequence by base pairing. After screening a genetic library with thousands of different messenger RNA sequences from jellyfish, he found one that bound just perfectly. To find the actual gene that produced the GFP, Prasher took a gel in which restriction fragments from the jellyfish genome had been separated and found that one of the fragments bound tightly to the mRNA. That fragment contained the actual gene for the GFP, and this method is called Southern blotting after its inventor, Edwin Southern. Today is often quicker and less expensive for scientists to search for genes in computer databases where the complete genomes of many organisms are available. So once a biologist finds a gene, a technique known as polymerase chain reaction, PCR, allows them to make many, many copies of it. A piece of DNA is heated, which separates its two strands. As you can see here, step 1. Step 2, at each end of the original piece of DNA, a biologist adds a short piece of DNA that complements a portion of the sequence. And these short pieces are known as primers because they prepare or prime a place for DNA polymerase to start working. Step 3, DNA polymerase copies the regions between the primers, and these copies then serve as templates to make more copies. And step 4, in this way, just a few dozens of replications can produce millions or billions of copies of the DNA between those primers. So how do scientists copy the DNA of living organisms? The first step is using the polymerase chain reaction method to copy a gene is to heat a piece of DNA, which separates its two strands. And then as the DNA cools, primers bind to the single strands. Next, DNA polymerase starts copying the region between the primers, and these copies can serve as templates to make still even more copies. All right, what is the importance of recombinant DNA? So scientists began wondering if it might be possible to change the DNA of a living cell, and many of them realized this had already been accomplished decades earlier in Frederick Griffin's bacterial transformation experiments. During transformation, a cell takes in DNA from outside the cell, and that added DNA becomes a component of the cell's own genome. Griffin's extract, or heat-killed bacteria, contained DNA fragments which were taken up by the live bacteria, transforming the live bacteria and thus changing their characteristics. Today, scientists can produce custom-built DNA molecules in the lab and then insert those molecules along with the genes they carry into your living cells. Machines known as DNA synthesizers are used to produce short pieces of DNA, up to several hundred bases in length, and these synthetic sequences can then be joined to natural sequences using DNA ligase or other enzymes that splice DNA together. A gene from one organism can be attached to the DNA of another organism. Restriction enzymes then cut DNA at specific sequences, producing sticky ends, which are single-stranded overhangs of DNA. If two DNA molecules are cut with the same restriction enzyme, here you go, their sticky ends will bound to a DNA fragment that has complementary base sequence. DNA ligase then joins the two fragments. The resulting molecules are called recombinant DNA, like recombination DNA. Recombinant DNA technology, joining together DNA from two or more sources, makes it possible to change the genetic composition of living organisms. By manipulating DNA in this way, scientists can investigate the structure and functions of genes. Scientists working with recombinant DNA soon discovered that many of the DNA molecules they tried to insert into host cells simply vanished because the cells often do not copy or replicate the added DNA. Today, scientists join recombinant DNA to another piece of DNA containing a replication or a start signal. This way, whenever the cell copies its own DNA, it then copies the recombinant DNA too. Pretty cool, isn't it? In addition to their own large chromosomes, some bacteria contain small circular DNA known as plasmids. Here we go, sorry. Joining DNA to a plasmid and then using the recombinant plasmid to transform the bacteria results in the replication of a newly added DNA along with the rest of the cell's genome. Plasmids used for genetic engineering typically contain a replication starting signal called the origin of replication, and a restriction enzyme cutting site, such as ECO-RI, as you can see right here. Plasmids are also found in yeast, which are single-celled eukaryotes that can be transformed with recombinant DNA as well. Biologists working with yeast can construct artificial chromosomes containing centromeres, telomeres, and replication start sites. These artificial chromosomes greatly simplify the process of introducing recombinant DNA into the yeast genome. Bacteria can be transformed by using recombinant plasmids. Scientists can insert a piece of DNA into a plasmid if both the plasmid and the DNA target have been cut by the same restriction enzymes to create the sticky ends. The new combination of genes is then returned to a bacterial cell. Here we go. Which replicates the recombinant DNA over and over and over again and produces human growth hormone. Ta-da! Very expensive stuff. The recombinant plasmid has a genetic marker, such as a gene for antibiotic resistance. A genetic marker is a gene that makes it possible to distinguish bacteria that carry the plasmid from those that don't. This plasmid contains the antibiotic resistance genes TETR, and AMPR. After transformation, the bacteria culture is treated with an antibiotic. Only those cells that have been transformed survive because only they carry the resistant genes. So our objectives describe the importance of recombinant DNA. So recombinant DNA technology, joining together DNA from two or more sources, makes it possible to change genetic composition of living organisms. Moving on to objective 3 then. Define transgenic and describe the usefulness of some transgenic organisms to humans. The universal nature of genetic code makes it possible to construct organisms that are transgenic, containing genes from other species. Transgenic organisms can be produced by the insertion of a recombinant DNA into the genome of a host organism. Like the bacterial plasmids we discussed, the DNA molecules used for transformation of plant and animal cells contain genetic markers that help scientists identify which cells have been transformed. Transgenic technology was perfected using mice in the 1980s. Big mouse, little mouse. Who wants bigger mice? I don't know. Genetic engineers can now produce transgenic plants, animals, and microorganisms. By examining the traits of genetic modified organisms, it is possible to learn about the function of that transferred gene. Be ethical to do this on humans? Not yet. Many plants also can be transformed using agrobacterium. In nature, this bacterium inserts a small DNA plasmid that produces tumors in a plant's cells. Scientists can deactivate the plasmid's tumor-producing gene and replace it with a piece of recombinant DNA. Recombinant plasmid can then be used to infect and transform plant cells. The transformed cells can be cultured to produce adult plants. There are other ways to produce transgenic plants as well. With their cell walls removed, plant cells in culture sometimes take up DNA on their own. DNA can also be injected directly into some cells. If transformation is successful, the recombinant DNA is integrated into one of the plant cell's chromosomes. Scientists can transform animal cells using some of the same techniques they use for plant cells. The egg cells in many animals are large enough that DNA can be injected directly into the nucleus. Once the DNA is in the nucleus, enzymes that are normally responsible for DNA repair and recombination may help insert the foreign DNA into the chromosomes of the injected cell. Recently, it has become possible to eliminate particular genes by constructing DNA molecules with two ends that will sometimes recombine with specific sequences in the host chromosome. And once they do recombine, the host gene normally found between those two sequences may be lost or specifically replaced with a new gene. This kind of gene replacement has made it possible to pinpoint the specific functions of genes in many organisms, including mice. Let's talk about cloning now. A clone is a member of a population of genetically identical cells produced from a single cell. The technique of cloning, uses a single cell from an adult organism to grow an entirely new individual that is genetically identical to the organism from which a cell was taken. Clones of animals were first produced in 1952 using amphibian tadpoles. Then in 1997, I remember this, Scottish scientist Ian Wilmot announced that he had produced a sheep called Dolly by cloning. Animal cloning uses a procedure called nuclear transplantation. The process combines an egg cell with a donor nucleus to produce an embryo. And you can look at this in a little more detail there. First, the nucleus of an unfertilized egg is removed. Here we go. Next, the egg cell is fused with a donor cell that contains a nucleus fused to the cell taken from the adult. The resulting diploid egg develops again into an embryo, which is then implanted in the uterine wall of the foster mother where it develops until birth. Cloned cows, pigs, mice, and even cats have been produced using similar techniques. Objective 3 is define transgenic and describe the usefulness of some transgenic organisms to humans. So transgenic organisms can be produced by the insertion of recombinant DNA into the genome of a host organisms. And the usefulness to humans is endless. Our objectives is to explain how scientists manipulate DNA, describe the importance of recombinant DNA, and define transgenic and describe the usefulness of some transgenic organisms to humans. Our goal, where is my goal? Oh, I don't have it down there, was to evaluate the impact of biotechnology on the individual, society, and the environment, including medical and ethical issues too. We'll talk about those in class. Enjoy.